All right, welcome to the next laboratory session. At the beginning of this lab session, we're actually going to take a short field trip to Victoria Harbor to collect 4 samples of water at different depths right by the central wastewater treatment facility. What fun. So after we collect those samples, we're going to bring them back to the lab and conduct a variety of different types of analyses. So in preparation for this lab, you should not only be looking at the lecture, you should be following this tutorial, and you should be looking through the protocol that will be posted later. So I have my protocol with me, and I'm going to first take you through the initial spectrophotometry assay, which half of the class will be doing on ammonium. And ammonium is a dissolved form of nitrogen that is common in marine and fresh waters, and it has a big impact on ecosystem productivity. In order to measure ammonium, we have to do some chemistry to exert a color change in a solution that can be detected by the spectrophotometer. And that color change is going to be proportional to the concentration in our sample. But in order to calibrate our chemistry with the spectrophotometer, we need to construct what's called a standard curve. Now, my demonstrators have already prepared the standards for you to generate a standard curve. These standards have a known stepwise increase in ammonium concentration, and you'll be able to see that the color change after the chemistry is conducted in each of those samples is going to be quite different. But to begin with, I need to get the reagents ready for the chemistry that can help us reveal that color change. And so the main tool I'm going to be using today is my micropipette. So again, if you don't know how to use one of these, please review the tutorial from last week on how to properly use a pipette. Pipetting on this class, for this section, is going to be really important. Now, you can see here the reagents and samples that we're going to be using today. So I have an alkaline reagent. I have a sodium hypochlorite reagent, which is essentially Clorox bleach. Now these two chemicals will be mixed together to create an oxidizing reagent that will be used in the final reaction. Additionally, we have phenol. Phenol is a somewhat noxious and toxic chemical, so you want to be sure that you're doing this chemistry inside of a fume hood to protect yourself. And then we have a nitroprusside solution that will be used in the chemistry as well. These two will be added directly to our sample. These two will first be mixed inside of an oxidizing reagent tube, so we're going to prepare that chemical fresh by mixing these two together just prior to incubating our samples. And right here in the front I have beakers that are filled with what for you will be your filtered samples, but for today I'm using our standards that we use in the standard curve in place of the samples. So right now I'm just constructing the standard curve to instruct you how to do this process. So the first step is that I need to transfer my standards into my reaction tubes. So over here I have 1, 2, 3, 4 open reaction tubes that are associated with the different concentrations that will be used to make my standard curve. And those concentrations are 0, 50, 100, and 200 micrograms per liter of ammonium. So I'm going to start from the lowest concentration and transfer up to the highest concentration, and that way I can conserve supplies. I can use the same tip for transferring those solutions, because there's no way a low concentration can influence a higher concentration. If I went in the other direction, I could cause big problems. So I'm going to transfer 2 microliters of each standard by pipetting slowly and using good technique. Okay, so I've transferred all of my standards into their reaction tubes, and now I need to start to prepare my oxidizing reagent using the alkaline reagent and the sodium hypochlorite, which is simply household bleach. So I'm going to use the tube that's supplied for me, labeled oxidizing reagent. I'm going to open it up, and using a new pipette tip, I'm going to obtain 4 milliliters of the alkaline reagent. So I've added 4 microliters of my alkaline reagent. I'm going to get a new pipette tip, and then I'm going to add 1 milliliter of the sodium hypochlorite solution. Remember to pipette slowly so that you don't get an inaccurate volume. So my oxidizing reagent is now prepared. I can give it a little mix and I can set it to the side so that it's ready for the next step. Alright, so I've mixed my oxidizing reagent. Now I'm going to add the phenol. And remember, the phenol is a slightly toxic, kind of a smelly chemical, so you'll be doing this all in a fume hood. And in order to add the appropriate volume of phenol for the first time, I'm going to have to change the volume of my micropipette. So instead of 1,000 microliters, I need to change this to 150 microliters. So, but to do so, I will spin this dial until I get to 150. There we go. So now I'm ready to add 0.15 milliliters of phenol to each sample. And again, as long as I don't stick the tip of my pipette into the sample itself, I can use the same pipette. And I'm going to want to cap the phenol right away so that I don't have to inhale the vapors anymore. And make sure that the aluminum foil is covering the phenol because it is sensitive to light. Once the phenol has been added, I'm going to quickly vortex each of my samples by putting the cap on tightly and touching it to the vortex plate. So I'm going to get a new pipette tip, and this time I'm adding 150 microliters or 0.15 milliliters of the nitroprusside solution, this stuff right here. And I'm going to add that to each sample. So once the nitroprusside solution is added, I need to do a quick mixing once again, and then I'm going to wait for 1 minute before adding the oxidizing reagent. Alright, the final step is adding the oxidizing reagent that we prepared. And the volume that's required is 0.37 milliliters, or 370 microliters. So I'm going to change the dial again. I'm going to increase the value to 370. There we go. And I'm going to add that volume to each of my reaction tubes. And once again, for the last time, I will put the cap on and I will vortex the sample. But this time, once I'm finished, I'm going to cover all of my tubes, the entire rack if I'd like, with some aluminum foil to protect the samples from light. Because light can alter the reaction and it can impact the color change that we see in our sample. So once I have my samples protected from the light, I'm simply going to wait 1 hour until the reaction has come to completion, and then I'll return to the station to analyze my samples using the spectrophotometer. So if you're assigned ammonium for your group, then you can get to this point and then you can move on to one of the next stations to continue to analyze your water samples for other parameters. Okay, station 2 is the phosphate station, and only half of the class will be doing this assay. Phosphate is another colorimetric assay. We're going to use chemistry to change the color of the solution based on the amount of phosphate that is dissolved in that water sample. So just like with the ammonium, I'm going to start by generating a standard curve based on dilutions of known quantities of phosphate that have already been prepared for you. But you need to do the chemistry and determine the concentration to create the standard curve from which you will determine the concentration of your unknown. So that's page 2 of the lab protocol. So some of the tools that you'll need the micropipette again. And again, please note that you will be required to change the volume of the pipette by turning this dial from time to time. We also have beakers here that you can use to organize your samples from the field as well as your standards. And then we have a variety of different reagents that are used to react with your water sample. The first one that we'll be using is a sulfuric acid, a 5-normal sulfuric acid solution, so you need to be careful with this. The second one is a potassium antimonial tartrate, which is another solution that is light sensitive, so it's been wrapped with aluminum foil. Another solution is ammonium molybdate, which is here, again light sensitive, so it's covered in foil. Please keep those reagents covered so that the next group can use them. And last but not least, we have ascorbic acid. The ascorbic acid is the key ingredient here. The method that we're using is actually called the ascorbic acid method for measuring phosphate in seawater and fresh water. Okay, so the first step is to create a combined reagent of all those chemicals, and we have labeled a beaker here for you with an empty falcon tube, and you'll fill this falcon tube with your combined reagent. And in an unusual twist, we're going to actually start with the sulfuric acid. So I'm going to open my falcon tube, I'm going to get a fresh pipette tip, and I need 5 milliliters of this solution, so I'm going to have to add 5 different aliquots, 5 different single 1 milliliter additions, carefully. The next step is to add 0.5 milliliters of the potassium antimonial tartrate solution. So I'm going to change the setting of my pipette to 500, which is 0.5 milliliters. I'm going to make sure to cap this and cover it so that it's protected from the light. Now I'm going to keep my pipette on this setting. I'm going to get a new pipette tip because I have to add 1.5 milliliters of ammonium molybdate. So I'll just add 3 squirts of a ½ milliliter each. Now I'll increase the volume of my pipette to 1 milliliter, and I'll add the final reagent, the ascorbic acid. So I add 3 additions of 1 milliliter. And that is the final ingredient of my combined reagent. I've added, using my pipette, 5 milliliters of each of my standard solutions to these reaction tubes that I've labeled and placed in this rack. And the next step is to add 0.8 milliliters, or 800 microliters, of my combined reagent to each of those reaction tubes. So I will get a new pipette tip, which have disappeared. And I'll now add 0.8 milliliters of combined reagent to each of my reaction tubes. 1, 2. Now I need to cap each of my sample tubes, and I will vortex it briefly, just like we did with ammonium. So now I'm simply going to wait 10 minutes for a color change to develop, and then I'll return. I'll transfer my samples into cuvettes, and we'll use the spectrophotometer to measure the absorbance. So we can see immediately that while we're waiting for the color change to develop, that it's obvious, and we're getting a chemical reaction that's converting our solution to a blue color. So in the blank, we don't see any color development. That's a good sign. That means that we truly have no phosphate in our blank. But in our 100, 200, and 400 micrograms per liter, you can see an increasing intensity of that blue color development. After our reaction has been completed, I'm going to transfer 2 milliliters of each of my standards and my sample into one of these disposable cuvettes. The cuvette can hold about 2 milliliters of solution, and it has special markings that will help you to orient its position inside the spectrophotometer. But first I need to add 2 milliliters to my cuvette, and I can do that by holding them both in one hand. That's 1. Oops. And that's 2. And this sample represents my 0 phosphate. So notice there's no color change here at all. But it does contain all of the other chemicals that I have added to the solution, including the combined reagent. So I'm going to place the cuvette inside of the spectrophotometer with this arrow facing me. So to do that, I'll first open the lid of the spectrophotometer. I'll remove a cuvette if there's one in there. And I'll place the cuvette with the arrow facing forward and push it all the way down. And then I'm going to close the lid because the spectrophotometer is using light. So if we have ambient light entering the spectrophotometer, it could affect our readings. Now the first step, because we know that this sample has 0 phosphate, we're going to tell the spectrophotometer that this is a 0 absorbance value. Even if the spectrophotometer is detecting some absorbance, we're going to adjust for that by pressing the zeroing button, which is here. So you can see the absorbance has been reset to 0. So now everything that we measure will be adjusted based on that correction. So I'm going to remove my sample, and I'm going to prepare the next one, which is the 100 micrograms per liter solution. And it's better if you hold the cuvette while you fill it so you don't tip it over. And I'll add that. And what you should notice right away is that the absorbance value is positive and much higher than the blank value of 0. So in my lab notebook or on my worksheet, I'll record this absorbance 0.197 as the absorbance value for the first standard of 100 micrograms per liter. And you will repeat this process for each of the standards as you complete the exercise. Once you have the standard curve values, you will input them into a computer, and you'll use the equation of the line from that computer to calculate the concentration of your unknown based on its absorbance. Okay, so this is the station for measuring salinity, and salinity is actually really easy because we have this handy little instrument called a refractometer. And a refractometer is simply a type of a scope that contains a prism that's mounted in the top. And the prism is actually itself going to bend light from the outside through this tube so that we can view it. And the degree at which that light is bent, or its refraction, is influenced by the salinity of the water that we're testing. So today we'll be measuring some seawater samples, so we expect the salinity to be much higher than fresh water. But just to show you how to use this, we open up the lid of the refractometer, and we can apply a little bit of water using an eyedropper or a wash bottle, or sometimes even just by dipping the refractometer into that solution. It's a pretty tough little instrument. But for demonstration, I'm just going to put a few drops of deionized water on the surface, just like this, and I'm going to slap the lid closed. So what you want to notice is that when the lid is closed, you have a thin layer of liquid covering the surface of that prism. Now, using a good light source, I'm going to hold that portion of the refractometer close, and I'm going to look through using my eye. And I'm looking for the interface between a dark blue area and the white area. And the line that is formed between them is indicating the salinity of the solution. And when I look through here, because I'm using deionized water, I can see that the number is around 0. If we expect to have some salt in this solution, we would expect that the line would increase and move up the scale. Now keep in mind when you are looking through the refractometer that on the right-hand side we are measuring in parts per thousand, and on the left-hand side we are measuring in specific gravity, which is milligrams per liter. If you find that the refractometer is out of focus, you can actually refocus it by twisting the end of the viewing piece here. So it doesn't matter if you're wearing glasses or have bad eyesight, you should be able to get the refractometer to a focus that suits your eye. Okay, the next station is also really easy. We're going to be measuring the pH of our solutions, and if you refer to the lecture you can learn a little bit more about pH. This is a thermoscientific pH probe. It's a pretty fancy excuse for a pretty simple piece of equipment. So you can see the main pH control unit. This is receiving all the data and converting it into a number that we can understand. There's a flexible arm that can position our pH probe. And this is the important piece right here. This is the pH electrode, which features a glass bulb at the end, which you have to be extremely careful with. So if you look closely, you can see that glass bulb, and it's surrounded on the inside and outside by an electrode solution. So you can see there's a liquid inside. So during this lab, you're going to be very careful not to smash the electrode bulb by treating this improperly. So you want to be very careful with the end of this probe. The other thing is that the probe will not give you good data unless you open up this vent. So there's a small cap that prevents the electrolyte solution inside from evaporating. But you have to remove that cap in order to take a good reading. So the cap must be removed while you're measuring, but once you're finished measuring, you must replace the cap and press it into place so that the solution inside remains at the same concentration that it's meant to be. So since I want to take a measurement, I can open up that vent. I can place my probe inside this arm just to keep it steady. And notice there is a liquid storage container where, when you're not using the probe, the bulb should be kept inside this solution so that it protects the integrity of the probe. But when I want to use it to take a reading from a sample, I can simply dip the probe into my sample and provide a little bit of swirling action. And I'm also careful to make sure that I'm not smashing the end of the bulb around. If you don't see anything happening on the instrument, there's nothing flashing or the numbers aren't moving, then you need to press the measure button to initiate the measuring function. So the pH value is going to be quite dynamic because water itself is dynamic, and even inside of a small beaker there's going to be subtle variations in the pH as you swirl the solution around. Hopefully you will reach some equilibrium where the numbers are not moving too much. But I would worry more about the tenths place. So here we're measuring 5.73. I would be more concerned about the 5.7, and those numbers are stable before you take a reading. If you wanted to be really careful, you would wait until this number stabilizes, but you could be waiting for quite some time. So for today's activity, let's be sure just to measure to the tenths place. So in this case, I might record 5.7, a pH value of 5.7. So now that I'm finished with my sample, I'm going to put the beaker down. I'm going to move my pH probe over top of this waste container, and I can rinse it with some deionized water, and that will prevent cross-contamination between samples. We can use these Kim wipes to gently wipe off the deionized water and blot the bottom of the glass bulb. Do not press at all on the glass bulb. And then you can return the pH probe back into its storage solution. Remember, if I'm finished for the day, I'm going to want to put the cap back on the vent on this side of the pH probe. If you have any issues with the calibration or you think your numbers are not accurate, please talk to one of your demonstrators. Okay, for the next two stations we're going to be looking at total suspended solids, and we're going to look at microbial indicators in our water. So each group, when you get to this point, will get this vacuum filtration rig. Now please keep in mind that the glassware in this tray is pretty expensive, so we need you to take extra care when working with this stuff. The first part of the vacuum filter rig is a sidearm Erlenmeyer flask. So it's a standard flask with about a 1 liter capacity, and what's important is that it has this special sidearm for applying a vacuum line. We also have this fritted glass filter. This is not what you're going to be filtering anything through without having a filter paper on top, but this allows the vacuum to be applied evenly to your water sample so that it's drawn down into the flask during the process of filtration. But remember, nothing should go through this without a membrane filter or a glass fiber filter on the surface, because if you embed this with particulate matter from your sample, it will be ruined. Okay? So this piece has a silicone stopper which can be fit perfectly into the top of the Erlenmeyer flask. We don't even have to press too hard, because when we apply the vacuum, it will be seated into that union. The next step is, the next piece we have is a funnel, and the funnel can hold about 300 milliliters of solution. And the funnel has a nice ground glass surface that forms a nice seal, especially when wet, above the fritted glass filter. But you'll notice that there's nothing holding this on here, so we have another piece, which is a clamp, a metal clamp, that you can squeeze to open up and slide over top of these two pieces to hold them tightly together. So this will keep your filter in place while you're conducting the filtration. It forms a nice seal between the glass pieces as well as between the silicone stopper and the flask, so that your vacuum is being applied strongly and evenly to the interface between the filter paper and the sample that you're going to add above it. So last but not least, I need to bring my vacuum to the flask, so I'm going to attach one end of my vacuum tube to the side arm here, and I just need to push it over at least one of those barbs, and then I'm going to add the other end to the vacuum line, which is the gray knob on the lab bench. And this can be a little tricky. Just keep pushing until you get that tube over the top. So now I can turn on the vacuum, and you can hear it. You can hear the vacuum being applied. And it's simply going to be sucking air from the room into the vacuum system until I add some solution to this vessel. But I don't want to do that because I don't have a filter in place. So I'm going to turn the vacuum off. I'll remove the clamp and reverse the process, take off the funnel so that I can apply a filter. For total suspended solids, you will begin with a 47 millimeter glass fiber filter. And I want to basically trap all of the suspended material in my water sample on the surface of this, and I'll weigh it using a balance. So here we have a balance, and in order to weigh or to calculate the weight of the suspended material, I need to first understand what is the mass of this filter by itself. So I'm going to add a plastic tray to the balance and close the door. And you'll see that it's now weighing the mass of my tray. Well, that's not what I'm interested in. So I'm going to press the tare button, and I'm going to zero the balance with the tray on top. And this way I can determine the mass of the filter by itself, which comes to 0.1312 grams. Easy. So now I'm going to take after I record that number, I can take the filter. I will place the rough side of the filter facing up on top of my fritted glass filter, the funnel that's on top of my Erlenmeyer flask, and I'll position it so that it's nice and centered. Now I'll return the funnel to the top, I'll clamp it down, and I can reinitiate my vacuum. Now for total suspended solids, you're going to be filtering 100 milliliters of liquid through this filter, and you want to be fairly precise, so you'll be measuring the volume using a graduated cylinder. But for the purpose of this demonstration, I'm just going to chuck some DI water in here so you can see what happens. So once I start adding my solution, it immediately is pulled through the flask by the vacuum. And when you have a very clean solution, it moves through the system very quickly. You want to be sure that if you're filtering a lot of liquid, that you do not exceed the level of the sidearm, otherwise you'll risk introducing liquids into our vacuum line, which will be a very expensive problem here in the School of Biological Sciences. So please turn off the vacuum and disconnect the tube if you need to empty the volume of this container. So in order to disassemble this, it might still have some vacuum pressure inside, so I'm just going to disconnect this sidearm tube, and you can hear the vacuum release. And then depending on what I need to do, if I just need to empty it, I can remove this whole apparatus from the silicone plunger or seal. And that's actually not moving right now for me, so I'm going to be more careful and conservative and disconnect everything from top to bottom. So I remove the funnel. I can use forceps to remove my filter very carefully. And then I'm going to place it into a labeled plastic tray that has my group number indicated on it. And I'll give it to my demonstrator for drying. Now, someone from your group must return in 24 hours to weigh the final mass of your glass fiber filter, and then use the difference between that and the initial value to calculate the total suspended solids within a volume of liquid. Ready? Okay, we're going to continue to use this vacuum filtration technique to help us understand what is the loading of microbes in the water samples we're taking. Today we're specifically interested in measuring Enterococcus, which is a group of fecal-associated microbes that are very abundant in the guts of mammals. And the most abundant mammals in Hong Kong just happen to be us. So we're going to be measuring the microbes that are coming out of our guts in our waste. So I have my vacuum filter apparatus here that I've just used to prepare the sample for total suspended solids. And since I've run some of that sample through here, I want to make sure that my system is sterile and I'm not introducing any extra microbes that are adhering to the surface of the upper part of my filter apparatus. So I'm going to connect everything together. And this is the only time you do not need to have a filter installed, because I'm going to add a little bit of very clean ethanol to the filter rig. So I'm going to pour the ethanol around and allow it to filter through. So this should sterilize the surfaces that are going to be encountered by my sterile membrane filter. Now, ethanol can also kill the bacteria that you're trying to grow on your plate. So in order to prevent that from happening, we're going to use deionized water, which should be sterile, to prevent that from happening. So we're going to rinse any of the excess ethanol through the system with a little bit of DI water. Now I'm going to shut off my vacuum, open up the filter rig, and get ready to capture some bacteria on the surface of a membrane filter. So unlike with total suspended solids, we're now using a membrane filter that's made out of a polymer, not out of a glass fiber. And you'll notice that each of these filters is wrapped individually in a sterile packaging. So there are no microbes or contaminants inside these packages. So I can tear off one of the filters, and I should have with me a pair of forceps. Now, there are other people in this group, in this class, who are going to be using these forceps for Enterococcus. So we're going to dip these inside ethanol to make sure that they're sterile. I'm going to peel apart the membrane, and you'll notice that the membrane filter has two sides to it. One side has a grid, and this grid is simply to help you with counting the colonies that grow on the surface. On the other side, it's white. So this side should be placed down onto the media in the plate that will be used to incubate the microbes. So I'm going to place with my forceps the membrane filter onto the surface of my fritted funnel. I'll replace the top of the funnel and clamp everything back together, and then turn on the vacuum. So for the purpose of this demonstration, I will simply measure a volume of deionized water, so we don't expect there to be any microbes in this water. And in fact, this is what we'll do as a group to have a negative control. We want to know that the Enterococcus are not just growing on the plates because of contamination, so we will test that by preparing this negative control. Now, the Enterococcus assay will be conducted with various dilutions because we actually don't know what to expect. We don't know how many microbes are there. There could be 0, there could be 20, there could be 12,000 of them per milliliter of seawater. So how do we determine what is the right amount of water to filter? So in this lab, you are going to take one sample from Victoria Harbor, which is assigned to you on the last sheet of the protocol, and you're going to be filtering 1, 5, and 100 milliliters of that solution, and you'll use the graduated cylinder to measure that. So for demonstration purposes, I'll just fill up a certain volume of deionized water as my control. I would make note of the volume and record that in my lab notebook, and then I will filter this slowly through my membrane filter. Very easy. Once the solution has passed through, the liquid has essentially been separated from my microbes. My microbes are now stuck on the surface of the membrane. And you can imagine them as single cells or single small groups of cells that will slowly grow on the surface of that membrane once I give them a proper environment in which to grow. So I'm going to take this apart again. And before I handle my membrane, I'm going to get my media plate ready. So we have these pink-colored Enterococcus specific media plates that are available for you to use. I'll open up the container and get one of the plates out. These small plates are the perfect size for the membrane filter, so the filter will fit perfectly in here. And you'll notice that there's two sides. There's a top and a bottom. The top has nothing in it, and the bottom has a thin layer of the pink Enterococcus agar. So the lid goes on just like that. So I'm going to remove the lid, and using my sterile forceps, I will pick up the filter carefully. And then I'm going to place the filter onto the surface of the plate. And I might need to very gingerly use my forceps to tap down on the membrane to make sure that there are no air bubbles underneath. If the membrane is not in contact with the agar, then it's very difficult for the microbes to access the nutrients in that agar that will diffuse up through the membrane. So now that I have the membrane on here, you can see it's not coming out. I can put the lid back on my plate, and then I'm going to label my plate and make sure it has my group number, the sample identification, and the dilution factor that I used. Did I filter 1, 5, or 100 milliliters through this membrane? So please keep track of all that information on your plates. And you can store these plates here in the classroom, and we will be taking them to an incubator to warm up to 37 degrees Celsius for 24 hours. So one member of your group will need to come back in 24 hours to check on these plates and provide a count of the number of colonies that you see. And to show you an example, here is a plate that is growing some Enterococcus. And you can see the reddish colonies that have grown up over a 24 to 48 hour period. And those are easily countable on the gridded membrane, and we can normalize them to 100 milliliters. So if this was the result of a 1 milliliter filtration, then we would multiply by 100 to get the number of colony forming units per 100 milliliters of that solution. And that's how you measure Enterococcus in seawater.